Okay, so in the last two videos we have looked at firstly the structure of the muscle, right the way from looking at it as the muscle itself as a bicep, and then we looked at how that is actually made up of a number of fascicles which contain muscle fibres. Each muscle fibre is effectively a muscle cell, and those muscle cells are surrounded by a membrane referred to as the sarcolemma, and within that you have got a series of long tubular organelles which are surrounded by the sarcoplasm and also by the sarcoplasmic reticulum. If you looked at an individual myofibril, so an individual organelle, that organelle is divided into a number of sections referred to as sarcomeres. We looked at the fact that the sarcomere itself has a number of different sections to it which are formed from the different fibres that are contained within it, both actin and myosin. And in the last video we then focused in upon that neuromuscular junction, and we looked at the arrival of an action potential at the neuromuscular junction, the depolarization of the motor end unit, and that then leading to the acetylcholine being released and binding with the ligand-gated receptors, resulting in an influx of sodium ions into your through your sarcolemma, depolarizing that sarcolemma, and then influxing sodium down through the T-tubules, those deep grooves within the sarcolemma, and that this influx down these T-tubules then results in the depolarization of your sarcoplasmic reticulum, which is the equivalent of the endoplasmic reticulum found in your myocyte, which surrounds the myofibrils. And within these there are calcium ions, and therefore when the sodium floods in, this action potential in the sarcoplasmic reticulum then results in the release of the calcium ions from the sarcoplasmic reticulum. So in this video we actually want to look at what that calcium is going to do next, and we need to come back to our actin and our myosin fibres in order to do that. So I'm actually going to zoom in to both of these fibres and show you the stages of contraction of your sarcomere as activated by the calcium once released from that sarcoplasmic reticulum. So the first component that we need to look at is the actin, and if we just remind ourselves of actin, so you looked at actin in module 2 when you were looking at your cytoskeleton, and we looked at it as a two peptide chains that were intertwined with each other. So I've drawn at the moment the two chains of your actin, so these two amino acid helixes that we are seeing, and these are actually referred shown here in the black. So this is your actin, okay, and I've drawn it in this way so that I can show you another molecule which is actually wound around your actin and plays quite an important role in your sliding filament theory here. So what you can see here is that you have got a troponin molecule, which is this molecule here, so troponin, and you have also then got wrapped around tropomyosin. And what you will notice is that at the state that we are in at the moment, your troponin is holding your tropomyosin in place. And at this point in time, when our muscle is relaxed, it's actually blocking your actin myosin binding sites. So here we have our actin myosin binding sites, and these are currently blocked. So at this point in time, there is no ability for those myosin heads to be able to bind with the actin, and that is because of this troponin is holding the tropomyosin in a position whereby it is blocking those actin-myosin binding sites. So the next part of the sarcomere that I wanted to look at was the myosin. So here I've drawn for you a myosin filament, and there are a few components of this filament that we just want to have a look at before we look at how the actin and myosin interact with each other. So the blue sort of main tubular here, this is the filament itself. So we'll just label that as your myosin filament. And then we want to also recognise that we have then got what is referred to as our myosin heads. And at the moment I'm showing this that attached to each of those myosin heads is a DP, so it then is in diphosphate. And this would be the situation at rest, and we will look at that later on. Now, one of the things to recognise here, a little bit of further information at this stage, is myosin as a protein is a type of protein that is often associated with motor proteins. It is a protein that converts chemical energy, so from ATP into mechanical energy, and we will look at how this is happening. So on the head itself, it has a component which is able to hydrolyze ATP to ADP and inorganic phosphate, and this would happen when it is activated by calcium 2 plus ions. So these are those ions that we have met previously that are going to be flooding out of the sarcoplasmic reticulum when an action potential reaches and depolarizes the sarcolemma. Now we have looked at all of these components, we now need to look at how these are working together. And there are three key stages to be aware of here. So the first of those is stimulation. The second is going to be the attachment phase. And then the third stage is the detachment phase. So we just need to look at what is happening in each of those phases. So in this first picture, what we can see is that in the resting state, the myosin is sitting alongside the actin, and at this point in time, the troponin is holding that tropomyosin across those binding sites, and therefore that myosin head at this point in time is unable to bind with the actin, and it is not possible to form those myosin actin cross bridges. At the point of an action potential's arrival, the action potential is going to depolarize the sarcolemma and then subsequently the sarcoplasmic reticulum. The voltage-gated calcium channels on the sarcoplasmic reticulum would then open, and that calcium is going to flood into our sarcoplasm, and it is going to then reach our troponin and bind with the troponin molecule. And what we need to do is look at what happens next. So we can introduce our Ca2+, and we then need to look at what is going to happen as a result of that. So in this second image, we can see that the calcium has bound with the troponin. Now, the troponin as a protein, once this has bound with this, is going to see a conformational change in its shape. And the conformational change is going to result in the pulling of that tropomyosin band along the actin filament. And the result of this, as we can see, is that we have now got exposed actin-myosin binding sites. Okay, so we are now able to allow the myosin to bind with the actin at the binding site and form cross bridges. And just to highlight where this is going to take place for you, in yellow you can see here this exposed site and where that binding is going to take place. And that's going to result in the formation of cross bridges between the actin and the myosin filaments. When the myosin head binds with the actin fibre, the binding results in a conformational change, And what we see as a result of this conformational change is that the ADP that was bound to that myosin head is now released. So the ADP is going to be released at this point of time of attachment taking place. At any time that there is a binding taking place, there is a conformational change which is going to happen as a result of this. And this is all part of that second stage of the attachment that is taking place. Once ADP has been released due to the conformational change caused by the cross bridges being formed, this then allows the opportunity for ATP to bind to that myosin head. Now, the result of the binding of the ATP is that we then see the conformational change again of that myosin head, and in doing so, that conformational change is going to lead to the release from our actin-myosin cross bridge that was formed. And I have just annotated there for you so you can see conformational change leading to the release of that myosin head. So in this final phase, we're now going to see the calcium is going to come and bind with the myosin head, and the calcium is then going to the activation of that ATPase element that we have mentioned previously. And in activating the ATPase, it is going to cause the hydrolysis of ATP to ADP and inorganic phosphate. So we're going to go to... So this hydrolysis of ATP is important because this is actually going to return the myosin filament, myosin head, to its original position. So it is really important here to recognise that this energy is not used to flex this filament and the myosin head, but it's actually used to return it to its resting state. Okay, just to give a very final, very quick overview of what is happening, so a summary of what we have looked at today, we want to note that when the action potential arrives at the sarcolemma, it's going to depolarize the sarcolemma and then subsequently depolarize the sarcoplasmic reticulum. This is going to result in the voltage-gated calcium channels opening. The calcium is going to diffuse through the sarcoplasm and it's going to bind with the troponin molecule. That is going to result in a conformational change in the troponin molecule, which is going to pull the tropomyosin along the actin filament and therefore reveal the actin-myosin binding sites. When this happens, the myosin head binds with the exposed binding site, forming a cross bridge. It results in a conformational change and therefore a flex of the myosin filament, which pulls the actin along and releases the ADP. The fact that the ADP has been released allows ATP to bind with the myosin head, which again then results in a conformational change and causes it to detach from the binding site. This then enables the calcium to bind and activate the ATPase component of the myosin head. That's going to result in the hydrolysis of ATP to ADP and inorganic phosphate. And the energy that is released in doing this is going to return that myosin head to its original position and resting position.